Okay, so we'll be discussing, I'm Rahul Sir, we'll be discussing the remaining parts of the biomolecule chapter. Okay, then. So, question number 16. The essential chemical components of many coenzymes are. So, we have question number 16. So, A carbohydrate, B vitamins, C proteins, and D nucleic acids. Okay, so what we have here is so question number 16, right? So, essential chemical compounds of many coenzymes are, okay, that is vitamins. So carbohydrates won't be doing it, okay, proteins won't be doing it, nucleic acids won't be doing it, because what is a coenzyme? So the thing is, a coenzyme means this is an enzyme, okay, and this is a substrate, and this is a special site on the enzyme, other site, to which the coenzyme will bind. The cofactor and the coenzymes will bind. The coenzymes are the vitamins, the cofactor are the minerals. And when they bind to it, then only what happens is the conformation of the enzyme changes, and then what happens is the enzyme, it will bind tightly to the substrate, to the substrate. So then it will be able to catalyze the reaction. Coming on to the question number 7, which of the following statements about enzyme is wrong? Okay, which of the following statements about enzyme is wrong? So what happens is, here you can see the four options. Enzymes are denatured at high temperatures. Enzymes are mostly proteins, but some are lipids also. Enzymes are highly specific. Enzymes require optimum pH temperature for maximum activity. So, in question number 17, what will happen is, you can say enzymes are mostly proteins, but fine. But all no, none of the enzymes are lipids. The other thing is, the option number B will be correct. And even if there are some enzymes which are not proteins, they are known as rRNA. Okay, like 23S rRNA in case of the prokaryotic system, which has repetitive transferase activity because of its specialized tertiary structure, right? So that is why ribosomes are known as ribozyme, but there is no special lipid that acts as an enzyme or can catalyze any reaction. Okay. So coming on to the next question, uridine present in rRNA. It is a nucleoside. Okay, question number 18. It is a nucleoside. So I'll draw the structure for you. Okay. So we have something known as uridine, uracil, then uridine, then uridenylate. Okay, these are there and specialized compounds. Okay, so uracil is a base, uridine is base plus sugar, uridenylate is base plus sugar plus phosphate. Means this is just a base. This means nucleoside base plus sugar. This is base plus sugar plus phosphate, so it's a nucleotide. Okay, so I'll draw the structure for you. Okay, So the structure that I'll be drawing is, this is known as your specific molecule known as uridine. And if you want to make it uridenylate or uridynylic acid, then you have to connect it with a phosphate group. Okay. So this is a uridenylate. That is the UMP, you can say. If you subtract the phosphate, it becomes uridine. Okay, that is the nucleoside. Okay, so now we'll be shifting our focus to the regular topics. Okay, so one of the regular topics that I'll be teaching you today, okay, is about, you can say, the human eye, you can say. So when we talk about human eye, okay, so in the last class we had talked about the specificities of human eye, right? So we had talked about the different structures present in the outside of the human eye, because we had talked about the eyelids. Okay, we had also talked about the eyelashes. Then we had talked about the various layers. Okay, so I have told you that in human eye, the inner layer, not the inner layer, the human eye is divided into three layers. I have taught you, right? The fibrous layer is the most outermost layer. Then we have the vascular layer, and then we have the specific layer known as the inner layer. So once we come to the fibrous layer, okay, anatomy of the eyeball we had discussed. It is 24, you can say, millimeter, almost an inch in diameter, and orbital fat will cushion and insulate the eye. Okay, so outer layer is the fibrous layer. Then we have the intermediate vascular layer or the uvea. Then we have the visual receptors or the photoreceptors located in the inner layer. Okay, so once we talk about the fibrous layer, okay, so the fibrous layer of the human eye, okay, this consists of, you can see, the outermost layer of the eyeball, okay, which is the whitish sclera and the transparent cornea. Okay, whitish sclera and transparent cornea. Okay, now what we have is the sclera. If we talk about, okay, the first part of the human eye, so the sclera, once we talk about, so the sclera or the white portion of the eye, it covers most of the ocular surface, covers most of the ocular surface. Now, when it does cover most of the ocular surface, so we should be pondering upon its function also, right? So, what is its function, you can say? So, its function is, okay, so the sclera consists, you can say, of dense fibrous connective tissue. Very, very important PMT question this is. It contains dense connective or dense connective fibrous tissue or dense fibrous connective tissue, you can call it any way you like, which contains both collagen and elastic fibers. Okay, rich in both collagen and elastin. Now, the layer is thickest over the posterior surface of the eye near the exit of the optic nerve. Okay, so the sclera is thinnest over the anterior surface, you can talk or you can call. It is thinnest, okay, over the anterior surface. Over the anterior surface. Now, the six intrinsic eye muscles they insert on the sclera, and what they do is they, you can say that there what happens is there is an amalgamation of the collagen fibers of the present in the skeletal muscles also, okay, covering the skeletal muscles, the, you can say endomysium, with those of the fibrous layers. Okay, now the surface of the sclera contains small blood vessels. Okay, the surface of the sclera also contains sclera contains small blood vessels. Okay, small blood vessels means which have nerves that penetrate the sclera to reach the internal structure. So they also contain nerve endings or nerves that penetrate, and since nerves are there, there must be the supply of small blood vessels, capillaries, which will be providing HbO2, means the oxygen and various nutrients to it, right? That penetrate the, you can say, sclera to reach the internal structures. Internal structures. Okay. So now about more of the things related to sclera is the network of small blood vessels which are interior to the bulbar region of the conjunctiva, which the bulbar conjunctiva is the conjunctiva that completely covers your, you can say, the internal eye region. Okay, that is the bulbar conjunctiva which covers, you can say, the anterior surface of the eye. Okay, and you have the palpebral conjunctiva which covers the inner surface of the eyelids. Okay, so the bulbar conjunctiva when we talk about, so the small network of blood vessels anterior to the bulbar conjunctiva, they generally do not contain enough blood to lend an obvious color to the sclera. Okay, so on close inception, however, the vessels are visible as red lines against the white background of collagen fibers. That is why they look white, because they do not contain high flow of blood. Okay. Now come to the cornea region. The cornea region of the fibrous layer. First we talked about sclera. Why it is white also, I answered that question, because it doesn't contain enough blood. Okay, or the blood flow rate is not that high. Then we come to cornea. Now the cornea is transparent. The transparent cornea, you can say, is structurally continuous with the sclera. Okay, it is transparent and is structurally continuous with the, you can say, sclera. Now The border between the sclera and the cornea is known as corneoscleral junction or corneal limbus. Okay, so the border between sclera and cornea is known as corneoscleral junction. In some books, you will also look at this to be referred to as the corneal limbus. A good comparative exam question. Corneal limbus also, corneoscleral junction also. Junction between the cornea and sclera, that is the corneoscleral junction. Now, underneath the delicate corneal epithelium, the epithelial cells making the cornea consists primarily of a dense matrix containing multiple layers of collagen fibers. Okay, it contains the connective tissue layer which contains not one but multiple layer of collagen fibers. You can write down deep to the delicate corneal epithelium. Epithelium. What we have is the cornea consists primarily of a you can say dense matrix which contains not one, not two, but multiple layers. Okay, dense matrix which contains multiple layers of collagen fibers. Okay. Now, they are organized so as to not interfere with the passage of light. So delicately they are organized. There are multiple layers, but they are organized in such a way. It's a great enthralling phenomenon of nature that they do not interfere with the, you can say, passage of light. So they are organized in such a way that they don't interfere with the passage of light, right? Next, the cornea also has no blood vessels. Make it very, very clear. The cornea does not have any blood vessels. No blood vessel supply. Okay, very, very important thing this is. Does not have any blood vessel supply. Okay. So it is, you can say, you can call it avascular, right? Avascular. There's no blood vessel, so it can be called avascular. Now, since it is avascular, one thing can be very, very certain about cornea, the transplantation from the immunological standpoint becomes quite easy, right? So its superficial epithelial cells, they obtain oxygen and nutrients, okay? its superficial epithelial cells, they obtain oxygen, you can say, and nutrients by a simple diffusion process from the tears. Okay, from the tears that flow across their free surfaces. That is again a great thing. And nutrients by diffusion through the tears. Okay, that is why tears are so very important. Not only lubrication but providing nutrient to the corneal epithelium. Okay, otherwise it will go for hypoxic degeneration, right? So hypoxia induced stress, ER stress, and free radical production, okay, oxidative stress you can call it, and then eventually the necrosis of the epithelial cells. So diffusion through tears, and tears come from where? The lacrimal glands, they produce tears, and tears that flow across their free surfaces. Across their free surfaces. Okay. So now we are talking about immunological standpoint or immunological perspective. Okay, related to this cornea. So we know that this cornea is a very very delicate structure. So damage to the cornea, okay, may cause blindness, even though the functional components of the eye may remain normal, like the photoreceptors. Suppose they are perfectly normal, okay, the rods, the cones, they are perfectly normal, okay. Even the, you can say, retinal ganglion cells, the intrinsically photosensitive retinal ganglion cells, and the rods and cones, these three types of photoreceptor cells, they are perfectly normal. Still, you can have blindness. Okay, you can develop blindness, you can become blind. So, damage to cornea, it's a very, very delicate structure, and damage to cornea can cause blindness. Okay, even if the photoreceptor cells are completely normal. Now, we talked about cornea not getting any blood vessel supply, right? What does it mean? The cornea has a very restricted ability to repair itself because it doesn't get blood vessel supply. So for this very reason, again, it's a disadvantage too, that corneal injuries must be treated immediately to prevent serious vision losses, because due to less blood supply, okay, less availability of nutrients, okay, only depends on diffusion. So cornea has restricted ability to repair itself. Okay. Then restoring vision becomes again quite easy in that case too, because we one can go for corneal transplant, but the thing is again, you need to have somebody dead or somebody willing to donate their eye, okay, eyeball, so that you can take the cornea out. Because even if it is an, you can say, allograft, same species but not the same individual, right? It's not an autograft where you have the same species and same individual transplanting, so you will be having human leukocyte, antigen, HLA minor and major completely matched. There will be no immune rejection, no graft versus host disease, okay, GVHT, nothing as such. But suppose you are going for an allograft, suppose I go for an allograft for a kidney transplantation. So after blood group matching, which is just a first minor step, we have to go for HLA matching, human leukocyte antigen, regarding the MSCs, major histocompatibility complex, class 2 molecules. So class 1, class 2 matching is done. Okay. So the thing is, even if HLA major matches HLA minor antigens, they never match. Okay. So they always are susceptible to the, you can say, destruction, first the recognition and destruction of the organ by the immune system of your own body, as soon as graft versus host disease. Because the graft is causing disease in the host, because the host immune system has been able to recognize the graft, and then it has mounted an immune offense on the graft or on the grafted organ, and thereby completely necrosing it and completely making it useless and biologically ineffective. Okay. So the thing is, suppose you got a kidney transplanted and your T cells in circulation, they are extremely, you can say, efficient in detecting self from non-self. So they can detect the antigens and they'll know that these are not the autoantigens, these are not my self antigens. So the thing is, they'll be then dividing, okay, into T helper cells, more and more TS17 and all. They'll be recruiting, they'll be secreting cytokines, and macrophages will be recruited, and complete, you can say, cytokine storm will occur. So the thing is, that is the most dreaded thing, and that's why we take, you can say, immunosuppressant drugs like muronomab, and you can say cyclosporin A. So those things can be taken to tame down the immune system. Again, the risk is not completely ablated, you can say like that. So sometimes even after taking immunosuppressant drugs, what we have is we have specific, again, graft versus host disease and the rejection. Okay, and immunosuppressant therapy for a long period of time, like muronomab, it will lead to you becoming more vulnerable to secondary infections. But that is not the case with respect to, you can say, the cornea. Okay, that is not the case. What happens is, in case of a corneal transplant, since it is devoid of any blood supply, so our T cells won't be able to reach cornea, right? Now, corneal replacement is quite possible, okay? Is probably, you can say, the most common form of transplant surgery, okay? So these transplants can be performed between the unrelated, completely unrelated individuals, where even HLA major matching has not been done. You don't need to go for human leukocyte antigen matching, okay? No HLA matching is required for cornea transplantation, as cornea is avascular. Therefore, T cells won't be penetrating it. They won't be able to recognize which antigens are present, whether it is self or non-self. Okay, so they will be having no issue of you can say, graft versus host disease or rejection, right? So the thing is, such transplants can be performed between unrelated individuals easily. Okay, so corneal grafts are obtained from where? From the donor eyes, I told you, but again, the person may be dead or should be able to donate his front portion or one of the eyes. Okay. So for best results, the tissues must be removed within 5 hours after the donor's death. Okay. The tissue must be removed within 5 hours of donor's death. So that is the case in with corneal transplantation. Okay. So now we come to the vascular layer or the uvea. Okay. Now the second layer of the eyeball, the intermediate layer, you can call it, the intermediate layer is known as the vascular layer or uvea. Okay. Now the vascular layer or uvea is a pigmented region which includes what? the iris, the ciliary body, and the choroid. Okay, is a pigmented layer. Pigmented layer that includes the iris, it includes ciliary body, and the choroid. So this is a very, very important comparative level or even home exam level question, that what comprises the specific vascular layer of or uvea, or what are the essential or prime components. Okay, what are the indispensable components pertaining to vascular layer or uvea? Therefore, your answer would be iris, ciliary body, and the choroid. Okay, these three. Then it also consists of, or it also comprises of, numerous blood vessels, also the lymphatic vessels. Okay, so it comprises of numerous blood vessels, also lymphatic vessels. Okay, there was a research paper where last year where they discovered the lymphatic vessels in the brain too. So that is known as the lymphatic vessels, right? So lymphatic vessels, and then it also contains the intrinsic smooth muscles of the eye, okay, which are involuntary, which we don't have voluntary control upon them. They are the smooth muscles, okay, tapering ends, okay. So intrinsic smooth muscles of the eye. So this middle layer, okay, this specific uvea layer has the following functions. The functions I'll tell you. So number one, it provides a route for blood vessels and lymphatics that supply the tissues of the eye. Very, very important function. Okay, otherwise there will be no route. So how will the eye get complete nutrition, right? So it provides, so we talk about the functions. It provides a route for blood vessels and lymphatics. Blood vessels and lymphatics that supply the tissues of the eye, that supply the tissues of the eye. What else could be its function? It regulates the amount of light that enters the eye. Okay, this will be explained via the lens concept later on in the lecture. So when I will be explaining the accommodation part and all, I'll explain this also, how the eye is like a camera and how the lens of the eye and how the aperture can be controlled. The lens is like a natural lens. The lens contains mostly the crystalline proteins. Okay, so it regulates the amount of light that can entering the human eyes. Because if this regulation goes wayward, we can have oxidative damage also, and the image protection may not be proper also. So that will be explained properly later down the lane. Now, third function is again very, very significant, that it secretes and reabsorbs the aqueous humor, okay, the soluble substance, okay, present between, you can say, the lens and the specific choroid, you can say, or the cornea only, okay. So that is, it secretes and reabsorbs the aqueous humor. Reabsorbs the aqueous humor. So any kind of ailments in the drainage of aqueous humor, because it is produced daily and it is drained also regularly, just like CSF. So any kind of ailment in the drainage or any kind of problems pertaining to the drainage of the aqueous humor leads to a disease known as glaucoma, where you have increased intraocular pressure, okay, because of the increased accumulation of aqueous humor, okay, and secretes and reabsorbs the aqueous humor that circulates within the chambers. That circulates within the chambers of the eye. So that is the, you can say, typical function of the middle layer uvea, okay. So we all have heard about aqueous humor and vitreous humor, okay. So aqueous humor is a, you can say, is a transparent watery fluid similar to plasma, okay, but it contains lower protein concentration. Very very clear, you should keep it in head, in your mind. And it is secreted from where? It is secreted from the ciliary body, which is a structure that supports the lens. I'll explain it to you completely. Now it fills both the anterior and posterior chambers of the eye, okay. And the vitreous humor is also there, but that is not drained out. And vitreous humor is located in the space between the lens and retina, okay. So that is there, the location of vitreous humor, okay. So the aqueous humor, if you check, its functions are: it prevents the eye dryness, maintains the refractive index, okay, and then it will provide, you can say, room for expansion of the cornea, okay, that is there. Or it will prevent also, you can say, the inflation of the cornea. And it has immunoglobulins, antibodies, which indicate role in the immune response against pathogen defense, or against the pathogen, to serve the defense purposes. And you can say it can also serve, in research it has been shown that it can also serve as a way to carry ascorbic acid or ascorbate in the anterior segment, which, and ascorbic acid acts as an antioxidant, or an, it will act as a free radical quencher, you can say. Now it will provide nutrition like amino acids and glucose to the avascular ocular tissues, okay, like the lens is avascular, the cornea is avascular, okay, the trabecular meshwork, and these are the avascular tissues, and maintains the intraocular pressure, okay, and causes inflation of the globe of the eye, okay. So those are the things regarding the aqueous humor that we should be knowing, okay. So the drainage problem causes a lot of issue, like it causes, you can say, glaucoma, normal tension glaucoma. So there are multiple types are there, okay. So that is present. IOP is there, intraocular pressure. It is called as, not inter, must be intra. So inside the eye, so intraocular pressure, okay, which will lead to increase in intraocular pressure occurs because of the imbalances in the creation and drainage of the aqueous humor, okay, which can lead to glaucoma, and then that can also lead to vision loss, okay. So that is there. So it lies between, I told you, the iris, the colored part of the eye, and the inner surface of the cornea, okay, that is there. So the posterior chamber, you can say, of the eye is completely filled with this aqueous humor, okay. And the next function of this is controls the shape of the lens. The vascular layer, it controls the shape of the lens, also controls the shape of the lens. So all this will be explained properly in the next lecture. Okay, controls the shape of the lens. Okay. So I hope you are able to comprehend the lecture, and if you had any kind of doubts or queries, you can definitely contact me on my personal number.